Good morning, students. Today in this lecture video, I will be discussing beta-lactam antibiotics. Beta-lactam antibiotics are the antibiotic agent that contain a beta-lactam ring in their molecular structure. This includes penicillin derivatives, penams, cephalosporins, cephans, monobactam and carbapenems. Most beta-lactam antibiotics work by inhibiting cell wall biosynthesis in the bacterial organism and are the most widely used group of antibiotics. Until 2003, when measured by sales, more than half of all commercially available antibiotics in use is your beta-lactam compounds. The first beta-lactam antibiotics discovered is penicillin was isolated from a rare variant of Penicillium notatum, since renamed as Penicillium chrysogenum. So, we are discussing beta-lactam group of antibiotics. So, beta-lactam group of antibiotics involves penicillin, cephalosporin, cefamycin. Why they are called beta-lactam group of antibiotics? Because they have a ring which is called as beta-lactam ring. We will discuss this in the structure. Together, the penicillin and the cephalosporin accounts for over 1/2 of all of the antibiotics produced worldwide. So moving on to the history of antibiotics. In 1929, Alexander Fleming characterized this antibiotic as antibacterial product of your fungus Penicillium chrysogenum called penicillin. He was working with Staphylococcus aureus, where all of a sudden he saw that there is a contamination of green mold. And to his astonishment, there was a zone of inhibition all around the green mold. And that zone of inhibition is due to the antibacterial activity of the compound produced by the Penicillium species. But he could not commercialize penicillin, so he was not recognized at that time. In 1930, even after the presence of the sulfur drugs, many of the microbial diseases were really uncontrollable. Then, in year 1939, Howard Florey and his colleague developed the large-scale process for production of penicillin. So you know that both Howard Florey and his colleague and Alexander Fleming got the Nobel Prize for discovery of penicillin. The new antibiotic was effective in your controlling various infections, namely staphylococcal and pneumococcal infections among military personnel at the World War II, and was effective for treating even the streptococcal infection more than that of your sulfur drugs. So penicillin G is active primarily against gram-positive bacteria. This is the simplest form of the penicillin, which was effective against gram-positive bacteria. But chemical modification of penicillin G structure, however, significantly changes the property of your resulting antibiotics depending upon the need. You get to see the structure of 6-amino penicillinic acid, which comprises of a thiazolidine ring and a beta-lactam ring. So this thiazolidine ring and beta-lactam ring makes your penicillin, and this R group basically varies from one type of penicillin to its various derivatives. This is a potassium or sodium salt of the penicillin. Moving on to our next slide, I told you that the structure of the penicillin and its derivative will vary depending upon the presence of the R group. So these are natural penicillin is this particular structure. Then comes the semisynthetic penicillin, which comprises of your methicillin and your ampicillin. So semisynthetic like ampicillin and carbonicillin are effective against even gram-negative bacteria. Penicillin G was not effective against your gram-negative bacteria as it was impermeable to the gram-negative bacterial outer membrane. So the structural difference in the N-acyl group, this particular group is your N-acyl group which I am talking about. This N-acyl group of this semisynthetic penicillin allows them to be transported inside the gram-negative outer membrane where they inhibit the cell wall synthesis. Oxacillin and methicillin are widely used beta-lactamase resistant semisynthetic penicillin. We will discuss beta-lactamase later in this module. So this is the mechanism of action of your beta-lactam group of antibiotics. So you get to see this is the structure of your bacterial peptidoglycan. Peptidoglycan monomers basically comprise of glycopeptide polymer, N-acetyl muramic acid, and the various amino acids, namely this green one is L-alanine, the blue one is D-glutamic acid, the sky blue one. The pink one is your L-lysine, whereas the red one is your D-alanine, whereas the deep blue one is your glycine. So with each monomer you get to see a pentaglycine bridge. So you get to see this two monomer with a pentaglycine bridge. Now comes the use of the enzyme transpeptidase, which is also called as penicillin binding protein. Penicillin binding protein will bring about transpeptidation, and once this transpeptidation is established, what will happen is that this will undergo the cross-linking between the monomers, and you will have a synthesis of your peptidoglycan, the cross-linking of the peptidoglycan monomer resulting in strengthening of the peptidoglycan cell wall. Transpeptidase reaction will basically release D-alanine and bring about your transpeptidation reaction. Now what does penicillin do? Penicillin, in short, inhibits transpeptidation reaction. Now the question is that how do they do this particular job? So beta-lactam antibiotic is the inhibitor of cell wall synthesis. An important feature is transpeptidation, the reaction that results in the cross-linking of two glycan-linked peptide chains. So I have shown you in my previous slide that there are glycan chains and there are transpeptidation between the two. The transpeptidase enzyme bind to penicillin, so-called penicillin binding protein. Why they are called penicillin binding protein? They are called penicillin binding protein because transpeptidase have a ability to bind with the penicillin. When PBPS binds penicillin, they cannot catalyze the transpeptidase reaction. So the transpeptidase reaction between the pentaglycine bridge will not occur between one pentaglycine bridge and a glycine alanine will not occur if there is this transpeptidase is inhibited by penicillin. As a result, newly synthesized bacterial cell is no longer cross-linked, and so they will not maintain the strength. In addition, antibiotic PBP complex stimulates the release of another enzyme called autolysin. The enzyme that digests the existing cell wall results in weakened self-degrading cell wall. So due to osmotic pressure difference, the cell can now lyse. By contrast, vancomycin, also a cell wall synthesis inhibitor, does not bind with the PBPS. So their mode of action is different, but directly binds to the terminal D-alanine peptide on peptidoglycan precursor, thus effectively blocks the transpeptidation. So from this slide you get to see the inhibition of transpeptidation in two different ways. So I will recapitulate the whole thing for you once. That is, we have two peptidoglycan monomers in the slide. These are the peptidoglycan monomers in the slide. Having N-acetylmuramic acid bound to 5 amino acid, namely L-alanine, D-glutamic acid, L-glycine and D-alanine. And there is a pentaglycine bridge which is attached to them, right? And then comes the use of transpeptidase. Transpeptidase is that particular enzyme which is also called as penicillin binding protein. They are penicillin binding protein because of a reason, because penicillin binds to transpeptidase. So before we move on to that, first let's see what transpeptidase does. Transpeptidase basically bring about the transpeptidation. You get to see this pentaglycine bridge binding with the D-alanine, and one D-alanine is being removed. And all the two peptidoglycan monomers are cross-linked by this reaction. So this reaction is very necessary for the synthesis and the strength of your peptidoglycan cell wall. But when it is your beta-lactam antibiotic, what does it do? Beta-lactam antibiotic is the inhibitor of cell wall synthesis. An important feature is your transpeptidation, the reaction that results in cross-linking of two glycan-linked peptides I have already told about. Transpeptidase binds to penicillin, so hence they are called as your penicillin binding protein. So when PBPS binds penicillin, they cannot catalyze transpeptidase reaction. But what will happen? That will result in stop to the cell wall synthesis. As a result, newly bacterial cell wall is no longer cross-linked and cannot maintain the strength and its rigidity. In addition, antibiotic PBP complex stimulates the release of another enzyme called autolysin, enzyme that digests the existing cell wall, results in weakened self-degrading cell wall. Due to osmotic pressure difference, the cell can lyse. By contrast, I will highlight another antibiotic, vancomycin, which is also a cell wall synthesis inhibitor. Does not bind to PBPS but directly binds to the terminal D-alanine peptide on the peptidoglycan precursor, thus effectively blocks transpeptidation. Moving on to the most important portion people are talking about nowadays is the mode of resistance. That means how microorganisms are gaining resistance against various antibiotics. So beta-lactam antibiotics, the most important structure is a beta-lactam ring. The effectiveness of this antibiotic relies on the ability to reach the PBP intact and their ability to reach PBP and bind to PBP. So if penicillin binding protein is binding to the penicillin, then only penicillin can block the transpeptidation, otherwise not. So what are the mechanisms which a microorganism can pose to become resistant to this antibiotic? The first one is enzymatic hydrolysis of beta-lactam ring. An enzyme is secreted by the bacterium and that is basically used to degrade the beta-lactam ring. Enzyme beta-lactamase or penicillinase is being produced. They hydrolyze the beta-lactam ring of the antibiotic, rendering antibiotic inactive. Example, if you want an example, it is the New Delhi metallo-beta-lactamase 1 discovered in 2009. So this is one of the superbug which was isolated in New Delhi, and it was found that particular superbug comprised of this New Delhi beta-lactamase, which is basically degrading the beta-lactam group of antibiotics present in bacterial chromosome or maybe acquired via plasmid. So this enzyme can be present in your bacterial chromosome or may be acquired by the plasmid. Now, this is a differential expression, right? Beta-lactamase gene expressed only when it is being induced by the presence of your beta-lactam group of antibiotics. So if you have beta-lactam, then that will induce the expression of this particular gene to produce beta-lactamase enzyme. We have solution to this problem. And the solution to this problem is your clavulanic acid. Here I have drawn the structure of clavulanic acid. And clavulanic acid is a beta-lactamase inhibitor. The clavulanic acid is designed in such a way to overwhelm all beta-lactamase enzyme, and thus it will effectively serve as an antagonist so that beta-lactam antibiotic is not affected by beta-lactamase. So the structure is such that they will act as an antagonist to the beta-lactamase and thus prevent the degradation of beta-lactam ring and making the antibiotic fruitful to the patient. There is another mechanism of resistance which I will talk about over here, that is your penicillin binding protein sometimes change their structure. So as they change their structure to a new novel structure, beta-lactam cannot, I will rather say your penicillin cannot bind to this particular novel structure, and so the transpeptidase enzyme remains your active. And as transpeptidase enzyme remains active, there will be continuous synthesis of peptidoglycan even if you have applied the antibiotic. Example of such pathogens are the evolution of MRSA, methicillin resistant Staphylococcus aureus. In my last class I have discussed that in 1959 methicillin is being produced to kill drug-resistant bacteria which was penicillin resistant. But now in 1960, the methicillin-resistant Staphylococcus aureus was developed. So it has developed in this way. We have also got in nature penicillin-resistant Streptococcus pneumoniae. So there are two ways by which they can pose a resistance to the antibiotic. One is by the production of beta-lactamase, another is by altering the penicillin binding proteins such that they do not react with your or do not bind with your penicillin. So with this, I will end my class today. We will start with a new antibiotic in our next class. So if you have any questions, if you have any queries, please do write in your Google Classroom, and moreover, stay home, stay safe.